Hi, Dr. Lillig. Thank you so
Hi.
much for being here today and letting us interview you. We are both with Chem Talk. My name is Roxanne, and we are interns on the podcast team. And right now, I'm a second year in human health and disease at Trinity College in Dublin, Ireland. And I'll let Claris introduce herself as well.
Hi, my name is Claris. I'm an incoming high school senior in Fremont, California.
Wow.
And Dr. Lillig, would you mind telling us just a little about yourself and your education and how you got here?
Sure. So my name is Jen Lillig, and I am a professor of chemistry at Sonoma State University. And right now I'm serving as the associate dean for undergraduate studies here. I've been at Sonoma State since 2003. Before that, I was a graduate student in biochemistry at UC San Diego. And then before that, I went to Harvey Med, where I got my bachelor's degree in chemistry. So haven't deviated much from the path.
Yeah. What initially got you interested in chemistry?
Honestly, it was my freshman chemistry teacher. When I went to college, I thought I was going to be an engineering major, not because I knew what engineers did or anything. Just seemed like the thing to do. But I really liked my first year chemistry teacher, and I ended up working in her research lab for a couple of years. And I figured if I could turn out maybe half as good as she did, I would be doing all right. So she had a really huge influence on my life.
That's amazing.
How did you know you wanted to become an educator?
I don't think I did. At Harvey Mudd, so once I switched into the chemistry degree, so I really liked my chemistry teacher, and then it turned out all of the other chemistry majors were people I really like to hang out with and like just be around and do homework with and things like that. And then most chemistry majors there went to graduate school. So I was like, okay, I'll go to graduate school. I didn't really necessarily know what graduate school was or what it entailed, other than it was about doing research. And if my advisor was recommending it, it seemed like the right decision to make. And so after I went to graduate school, I learned some things I didn't want to do. I really liked working in the lab, but I didn't want it to be the only thing that I was doing. I really liked being a teaching assistant, which was a requirement for graduate school. And then while I was there, right about the time in my fifth year when it was time for me to start thinking about graduating, the University of California started this faculty fellows program where you could teach part-time at the University of California and you could do research, postdoctoral research in somebody's lab. And so it let me do both. And then I was hooked. Like I could be a full-time instructor and teach classes. And then I got to do research in a lab that was totally different than what I did as an undergrad. And it had different types of pressure, associated with it. So my whole career wasn't depending on whether or not one experiment worked, I guess. So once I could do that, I found out that's what I wanted to do. I wanted to work with undergraduates. So that's how I ended up at Sonoma State.
That's great. And do you like it there?
Oh, I love it.
Yeah.
I absolutely love it. I feel like it's a really good environment. I love my professor colleagues from across the university, not just chemistry, but I have friends in all of the schools and all of the departments. And I think being an associate dean now has really helped me appreciate a lot of the other majors as well. We're a liberal arts university. And so it's really fun to look at and work with the different departments to see how we integrate the arts and the humanities and the social sciences and the sciences, and how all of those things come together to sort of make a sort of holistic education for students.
So that's primarily then what you're doing as an associate dean, is integrating between the different parts of the school?
Yeah, I kind of do two things. So I've been in this position, this is the end of my second year. And so before, right in my, before I transitioned into this, I had just gotten a chemistry grant with some colleagues at Sonoma State, some colleagues from University of California, Berkeley, and some colleagues from two community colleges up here, Diablo Valley and College of Marin, to work on student sense of belonging in STEM. Because as you know, it is not a diverse field in chemistry or STEM overall. And this grant was curricular in nature. So really focusing on developing curriculum for professors that would really support student sense of belonging and different sort of gateways into science, focusing on introductory chemistry. And at the same time, I was still running a lab. I haven't had students this past year, but my last student just graduated, where we studied antibiotic molecules, so that kill Listeria Monocytogenes, which is a food-borne pathogen. So we would look at how those molecules knew which bacteria to kill and which parts of the molecule were important in that killing activity. So sort of two things going on at once as I've sort of transitioned.
So I know you were talking about how you developed curriculum.
Mm-hmm.
Can you talk more about how you kind of integrate this into your daily life as a professor?
Yeah, so, it's interesting when you go to graduate school, they don't teach you how to teach chemistry. That's not the focus. And then you go get a professor job and you teach chemistry. So in the curriculum development, there are a lot of ways you can teach chemistry. You can sort of teach it the way that you learned it, which was somebody got up at the front of the classroom and lectured, and your students took vigorous notes, and you had 3 midterms and a final, and that was it. It's not a very inclusive way to learn chemistry. And there are a lot of other high impact practices that support it better. So the curriculum that we've developed, we're called the Chem Avengers, gave ourselves a name and made a logo. This curriculum has different entry points. So you have students working in teams, and they can look for patterns in data. So some of those patterns might be numerical. Some of them might be colors, like they can see changes in colors, and they can enter and participate in the analysis of data, which is what scientists and chemists do. We don't just go take tests, right? Like you do in Gen Chem. We don't do that. So it's teaching the chemistry in a way that better models what scientists actually do on a day-to-day basis. And so once students realize that they can contribute in these different ways, it's more confidence building, and then it can kind of go from there. It also, I think, entails a lot of self-reflection about what students should really learn in chemistry and in college, right? Like I have to look back at how much chemistry did I really know walking out the door of my degree. And I mean, I knew some stuff, but I didn't have like this Rolodex of facts in my head, right? Yeah. So it's really trying to be thoughtful and reflective so that students have more critical thinking skills and can apply their science, and that will help them learn the material sort of by default.
That's so interesting. And I really like that, because I was one of those students that I really struggled in chemistry. And I think a lot of it was because, I would take these tests and then realize like it was all gone. Mm-hmm. And I didn't remember anything. So I think the push to start developing curriculum that has it kind of stick with you is such a good thing that's happening.
And it's really hard as an instructor, right? It's not an easy transition because, from my group in college, right, we all learned one way, and it's natural to teach the way that you learned it. And so it gets really hard. It can be really hard to really sit down and reflect about what am I doing and what can I do better, right? I think it's really easy at first to be like, well, they just didn't study for the test, or nobody's asking any questions. Nobody did the reading, right? Which is what other people are doing.
Yeah.
But as an instructor, you can really only focus on what you can change. And so those can be really hard conversations to have with oneself, as you might imagine. So long process.
Yeah. So you mentioned that you've been studying Listeria.
Mm-hmm.
So that's a bacteria, correct?
Correct.
Do you mind kind of explaining what that is, how it fits into the realm of bacteria in general, and why you think it's important to study?
Yeah, so Listeria monocytogenes is a food-borne pathogen. So a lot of times you hear in the news, right, that there's been a food recall. So it's either Salmonella, E. coli, or Listeria. And back when I was starting here at Sonoma State, there had been a variety of food recalls. One of the biggest ones was I think it was cantaloupe recalls in Colorado. And a lot of people died. As far as pathogens go, though, it can't really make you that sick if you're healthy. But if you're immunocompromised or you're pregnant, Listeria can be dangerous. So the lab where I postdoced when I was doing the part-time teaching and part-time research was a pathogen lab, and they did a lot of really big Listeria projects, like looking at big proteins and how Listeria invades the immune system and things like that. My training before that was on small peptides, small proteins, and how they work. And so when I was looking for what I could do at Sonoma State, which is an undergraduate institution, very limited in equipment, that types of things, I started just going through all the papers published on peptides and Listeria. And I found a class of molecules, it turns out, that target Listeria and prevent it from invading, essentially. So it kills it off. And so what we do is we study how these molecules, like what part of the molecule is important for killing this Listeria. So you can mix it with healthy cells or other bacteria and it won't do anything. It only targets the Listeria. And then we study how does it kill it, right? So what parts of the molecule stick to the surface of the Listeria, and essentially it pokes a hole in the Listeria membrane, and then, all the Listeria innards spill out, essentially, for a prokaryote. But yeah.
That's really interesting. How does it normally invade the immune system? Like what, because I know, there's, in terms of bacteria, they all have different like parts of them that make it really useful for infecting whatever they're going to infect. So what is it about Listeria that makes us really good hosts?
I mean, it's got some really tricky molecules inside of it. So, your cellular defense systems will come and engulf the Listeria, right? So normally, we have cells that engulf the Listeria and then break it down by making a really acidic environment, right? But what happens with Listeria is when it gets engulfed and the environment gets really acidic, it has this molecule that all of a sudden like sticks a leg out. Not a leg, it's not really a leg, obviously, but it sticks a new piece of molecule out that lets the Listeria bust out of the vacuole that was intended to kill it. So it can escape our immune defenses. It has amino acids that aren't recognized by our immune system. So it's a tricky little bugger.
Yeah. And how did you discover what part of the molecules that you guys were using to kill it? Like, what's the relationship there?
It's interesting. So the really cool thing about these molecules, they're called bacteriocins, and they're all very similar. So they have these molecules are made up of amino acids, and the sequence of the amino acids matters. And so, but you can have like 10 bacteriocins, and they all have some parts that are very much the same and some parts that are very different. And so the literature on it, though, will show that like sometimes it's the front end of the molecule that tells the molecule which bacteria to kill. And then the second half of the molecule is the part that does the killing. So what we would do is, since I'm a chemist, is these are small molecules, small proteins. So we can actually synthesize just the front, and we can synthesize just the back separately. And then we can add them together and see if they can function as the whole molecule did. And one thing that we found, which was pretty surprising, was that if you chop the molecule in half, both halves can actually kill on their own. So we didn't expect that at all. I mean, these are not big molecules. You'd think if you chopped it in half, it would be dead. But it wasn't. And so, I mean, they definitely don't kill as well as the full-length molecule on its own, but they could both stick to the Listeria and they could both kill it. So that was sort of our biggest thing that we found as a lab group. And that was done by undergraduates.
Wow.
So yeah, all the work in my lab was done or has been done by undergraduate students. And actually that one, part of that project, I mean, multiple students worked on it, but part of that project was actually, the idea came from the undergraduate student. It's like, well, I just want to make this part of the molecule and check it out. I'm like, okay, that sounds good. Let's do it. And lo and behold, I didn't think it was going to work.
Yes, I wouldn't have thought that either.
I was like, if you really want to, we can do that, because I wanted to support what they were working on, because it was about them. To me, the research was about the experience the students were getting. But yeah, that was pretty cool. A lot of science works out like that. Very serendipitous.
Wow.
Yeah.
I guess, are there any challenges or limitations to your research, and how does like one day like in the lab look like for you?
So challenges, one thing about being a biochemist is that you are at the whim of the organism that you work with sometimes. So, Listeria grow at a certain rate. And so in order to do our experiments, for example, where we would measure, how well our molecule was killing the Listeria, you have to grow them overnight, and you have to grow them for another 15 hours, and you have to grow them for another, and then you can do the experiment, which takes 2 hours. And you can imagine, as an undergraduate student, you've got 3 different classes that you're trying to fit in. So just the planning, to execute an experiment is pretty hard for an undergraduate, just from a timing sense, let alone all the boo-boos that happen naturally in lab. So that piece was challenging. I think it's also hard in general. It takes a long time to do science. It's like you don't just walk into the lab and do an experiment and you get an answer. It takes years often to set up an experiment and get it to work and get all the bugs out and things like that. And, undergrads don't have that kind of time. A lot of times they won't, find you in the lab to join a lab until juniors or seniors. And by the time they get up and running, they graduate. And then they leave. And so a lot of starting over. But certainly sometimes, like the last student I had, he's at UCLA now, and he started as a freshman. And he worked in my lab for, he came at the end of his first year, so 3.5 years. He got a lot done. So if you're thinking about that, I think that's really important. The sooner you can get into a research lab and sort of get that experience, I think the better it is. Plus, it's totally different. Even if you have a really progressive chemistry professors, right, that are doing all of these cool things to help learning, what you do in class is totally not the same as what it's like being in a research lab. There's often no right answer, and sometimes, there's no answer at all. Just more questions.
Yeah. What was that student working on?
He was working on which pieces of the molecule.
Okay.
Yeah. And I had some students, we had a lot of different variations of those experiments going, because to overcome some of the challenges I would have, I usually have 6 or 7 students who worked in my lab at any given time, not physically, but over the semester. And so they would all be doing the same type of experiment, but with different combinations of, the front and the back of the molecule, or the whole molecule, or a different bacterial target, right? We were trying to see, could we also change the molecule and make it target a different type of bacteria. And so he would work on various aspects of that because he was there for so long, I could keep moving him to different projects.
And did it turn out that if you change the molecule, it does target?
Oh, that part we didn't get done yet.
And do you ever, like, when they graduate or move on, do you ever finish, sort of start to finish what they were doing, or how does that work?
It's tough. I say that the work that gets finished the most is the work that students are writing up as they go along. Over the course, when I first started working at Sonoma State in 2003, I had a very, completely unrealistic expectation about how this was going to work. Right. And so, the best projects that get completed are the ones where students kept meticulous lab notebooks. Because then I can pick up the lab notebook and I can be like, okay, or I can take the next student and be like, this is where we left off. So this projects that got finished were projects like that, where the students were really involved in the work, which means they were really involved, like communicating with me. And I could pick up their lab notebook, we would go through it, and I could talk to the next student. But sometimes, students find out that they don't like research. It doesn't have an answer that you can put a box around. And that wasn't satisfying, which is fine. Right? Different, it's good to get the experience and be like, nope, that's not it.
Yeah.
So those, I spent a lot of time starting over. Because then I'd look in the lab notebook too and be like, yeah, I don't know what they did. And you think it's going to be clear. Even my own lab notebooks, I'm like, oh, yeah. And then, later I'm like, what in the world am I talking about?
Funny. So when it comes to having to start over a lot of times, where are you in your research with this when it comes to, how much of this molecule do you have to target the Listeria and kill it? Like, where are you at this moment?
So at this point, what we've shown is that you can break it in half, and that the full length is the most effective at killing Listeria. The second half of the molecule, the back end, is second best at killing the bacteria, the Listeria, and the front end is the third best, but significantly less. And so the next step for that we were working on was what happens, can we recover the full activity if we add the two molecules separately? So we would add the synthesized front half, the synthesized back half, and then measure the activity. That's the next experiment.
Interesting. And what's that molecule called?
It's a bacteriocin. The one that we're studying is called piscicocin V1A and V1B.
Okay.
So, yeah. And so that one in particular, there's two of them. And these are made, I guess I should backtrack a little bit. These molecules are secreted by other bacteria in the environment so that they can outcompete other competing microbes for resources, right? So this, it's made by a bacteria called Carnobacteria piscicola. So C. piscicola secretes these molecules, they go out and kill Listeria, and they secrete a whole bunch of other ones too that kill other things, and then C. piscicola can thrive.
Interesting.
Yeah.
And then would you say you ate some contaminated food, and so now the Listeria is happily inside you. Would you be taking this molecule as like an injection or a pill? What are your thoughts on that?
So you probably can take it as a pill because it's a protein. It'll get broken down in your digestive system. I think the biotech sort of applicable use would be more of a preemptive. So you could sprinkle it on food so they could use it as like a food. I don't want to say food preservative because that implies something else.
Right.
But these are molecules that are known to be safe because you eat them every day. Right. So you could put it on food and prevent Listeria from growing in the first place.
So it would,
I think that's
a long shelf life then?
Yeah, they're super stable because they're small. Yeah. Was another reason why I picked working with them, because since we have such high turnover with our undergraduates, you can leave it on the lab bench. It's not light sensitive. It's heat stable. It's great.
So if you were, at like packing facilities, is that maybe where they would start with it and it would kill it and then prevent future.
Yeah, something like that.
Okay.
And there are similar molecules like this already out there, and like Neosporin and things like that. Those are completely different classes. Like there are all sorts of different classes based on what the molecular structure looks like. So some are peptides, like the ones we study. Some are two peptides that work together. Some are round, they're cyclic. So lots of different classes.
So in terms of bacteria as a whole, as you've said, there's lots of different shapes and there's lots of different stuff on them, like that help them move around and enter cells. In terms of our immune system and how it enters, what are the specific components that allow it to enter our cells?
You mean like what are the specific molecules?
Yeah, like what's on the surface? Because I know a lot of them have things on the surface that can kind of help enter our cells.
Yeah, so Listeria has a cool one. What's it called? I think it's called internalin. And what it does is it will secrete this molecule into its environment, right? And this molecule, when it gets close to its target cell, right? So the cell in your body, it causes that cell to ruffle.
Okay.
And then to envelop it.
Okay. So when you say ruffle, it's what you would imagine.
Yep. It's what kind of creates a ruffle and then it engulfs it.
You wanted to study this because you'd seen literature on it before and you thought this would be good for where you're working.
Yeah, I mean, I feel like I got really lucky in finding it. So my graduate work was on these peptides that associate with cell membranes and how do they do that. My postdoc work was on Listeria and HIV and pathogens. So I've always been into that, right? Like these disease-causing microbes. And then I swear I was going through you can do these database searches, and I was just trying to go through and find something that might merge those two things and be doable at Sonoma State. And I think I got to like number 65 or 70 in terms of article names. I like, searching membrane peptides and pathogens or something like that. And then I found one about, this bacteriocin, kills Listeria. And I looked it up and I'm like, this is perfect. It sort of meshes my two expertise. Mm-hmm. And I mean, the other good thing about it was most of the work being done in the field was being done in terms of like discovering these molecules. Mm-hmm. So it's like, we had this milk product with this bacteria and it makes this molecule. We found another one. I mean, they would find so many that sometimes it would be the exact same molecule, but found in two different organisms. So it has two different names.
Okay.
But what I do as a chemist is I study how does it work, mm-hmm. right? What parts of the molecule are important for what this does? And there aren't that many people doing that.
Right.
Which means on the time frame of undergraduate research, I wouldn't have to worry about, we call it getting scooped, right? Whereas a big lab at, MIT was like, we solved this problem.
Okay.
Before I could even get off the ground.
I see. So it's something you can kind of rely on to be there for you to study in.
Yeah, and to make a valuable contribution or a contribution, right?
Yeah.
So I think that's important that, if you're an undergraduate and you start a project and then the next month it's like, sorry, these people at MIT finished it. We're going to do something else now. That would suck.
Yeah.
It would suck for me too.
Yeah.
Yeah. So the rate of progress in the part of the field that I can contribute to was good for Sonoma State.
Yeah. Then that sounds like a perfect topic for you and your students to be able to.
Yeah, I think it's been pretty popular. Like I think students like, disease causing things.
Because you get to see like a real life display of what it's doing.
Yeah, exactly.
That's why I think disease stuff is cool, because you can see it so widely among people and then you can trace it back and see exactly where it came from.
Oh yeah, and it's in the news, so they're sort of familiar with it. Plus, like, I think it blends the biology aspect in really well for students that like both. And so I would sometimes, I've had biology students work in my lab. I've actually had kinesiology students, psychology majors. And so there are different parts of the project based on, are you more chemical minded? Are you more biological? So I had some students doing DNA work, right, to make mutations in the gene of the protein so that we could study the mutants. So, and then, a chemist would take the mutant and go do something with it. So lots of different, I guess, entry points, depending upon the interests of the students.
Yeah. What were the mutants doing?
So it was cool. We made some mutants that there had been some, wait, have they studied it yet? We picked different amino acids that were thought to be really important in either how the molecule targets Listeria or how it binds to the membrane. Mm-hmm. And so there was one in particular where we were able to change it. It wouldn't stick to the membrane at all. So we could look at, the change in one amino acid and how it changes the orientation of the peptide when it's bound to the surface. So you can imagine if this is your cell membrane and you have this molecule that comes down and lays flat, then it kind of just sort of pushes on the membrane and makes it leaky, and stuff can kind of come out and kill the Listeria. Mm-hmm. But if you have if your Listeria is flat and your peptide comes in like this and sticks a hole all the way through, that's much more effective at killing. Okay. So we could see differences with the mutation.
So some of those mutations clearly made it more effective at killing. Did some of them render it completely ineffective?
Yeah. There was one in particular, and then that changed the polarity of the amino acid. So it basically put a really hydrophilic amino acid in a position in the molecule that would go all the way through the membrane, which is hydrophobic. Mm-hmm. Hydrophilic and hydrophobic don't go together. Mm-hmm. And so that particular mutation, there was also evidence in the literature that was theoretical. So they would model it with a big computer simulation. So we were able to see the similar results in the lab.
Wow, that's so interesting.
Yeah, that was fun work. That was actually a master's student from the biology department who worked on that.
Okay. And you had mentioned that there was a kinesiology student in the lab. Mm-hmm. What was their role?
He had a project.
Okay.
Yeah. I could take anybody into the lab regardless of background. So you didn't need to know anything. Like, my perspective is that you can learn what you need to learn as you go. Like, it's not that you need to have some level of general chemistry or organic chemistry first. So this student that I had, the kinesiology student I'm thinking of, I had as a junior in my biochemistry courses. Mm-hmm. And he's like, "Can I work in your lab?" I'm like, "Sure." So he worked there for 2 years.
That's cool. Yeah. Was he working on Listeria?
Yeah, he had a project.
Okay.
So and so the students would teach each other different types of we sort of had like our sort of cookbook of experiments that we would do. So, the older students that were farther along would teach them how to do the experiments. Same thing for the psychology major. A lot of these students that I did have, even if they weren't a chemist or a biologist, were somehow pre-health oriented. Mm-hmm. So like for a while, the kinesiology student was interested in going to medical school. The nursing or the psychology student ended up getting their master's degree in nursing. So, okay. But yeah, I could take anybody. I mean, the most important thing to me was for people to be interested. Mm-hmm. It's hard it's hard. I could see students that are doing it because they felt like they had to do it. And that's no fun for anybody.
It probably doesn't get great results either if you're not interested.
Doesn't get very far. And, every research advisor is different. Right. And so some are very, micromanage, right? Like they're in the lab next to you. What are you doing? We're going to do this or do X, Y, and Z. I am not like that. I am more like, my door is open. You can come anytime you want. But if nobody comes to see me, I'll forget that you work here. Right. So, and I tell students that before, I'm like, really think about what type of mentorship do you want? I'm like, if you want mentorship and you're like, I need to talk to you, I'll talk to you for hours. And, I had students that would do that. They'd be like, I'm thinking about doing this experiment, or can you come in the lab and show me this? And those were always the most effective, like in terms of moving the projects forward. And I think in helping the student figure out what their next step was going to be like after graduation, which is a big part of the mentorship.
Yeah, that support just seems so crucial to know that someone is there to help you and want you to succeed.
And you get them for life. It's not like, they graduated. I'm like, see ya. I still talk to them. I follow them on Facebook if anybody ever uses that anymore. I don't know. Apparently not even Twitter. I don't know. But, they'll send emails or letters of rec, or I'm thinking about doing this. So I love keeping in touch with my students and seeing them go on to do whatever it is that makes them happy. It doesn't have to be chemistry, right? A lot of times it's something else entirely, and that's fine, right? That's part of the process.
Yeah, that's great.
I had a question going back to the curriculum you developed that we talked about earlier. How do you foster that kind of learning environment in the lab? And like, how do you make sure your students know that one, you're there for them, but two, they also have to push themselves to kind of, take initiative and learn what they need to learn in order to succeed?
I think probably it might be really hard for the students, and I try and make sure they understand this, but I will let them fail when I know that it's going to fail, right? As long as we're not getting ready to break like a really expensive piece of equipment or waste like a really expensive, reagent. I think failure is the natural part of the experimental process and the learning process. So I think that's a big piece of it. So at least I've always tried to make sure my students know that look, you're going to go in here, I'm glad you think you're going to finish this in 16 weeks, but I'll be happy if we get to step 2. Right. So to give them perspective about this is, the reality of how fast this is going to move. And a lot of times I'll tell them like, I'm not going to tell you how to do this. I need you to like sit down and make a plan. Then we'll go over the plan. I'll give you some feedback. And eventually you're just going to have to go in the lab and try it out. And so I think that that's really, really important, because if you go into chemistry and you haven't practiced failing, then I think it can be very hard on people. And so like I fail a lot. I mean, I have a whole book on failure, right? That's what we do. You fail most of the time until you have that one piece that moves forward. And so I think that's one of the most important parts, is preparing my students for that, and that it's okay, and that we celebrate the small victories too. Like, oh, look, you grew you made your first plate of bacteria and they grew. Yes. Right? Or you poured your first set of plates to grow the bacteria, even better, right? They solidified winning. We're winning.
Yes.
Right. So I think that that's part of it. And to also help students move from the focus on what the answer is to more the focus on the question and the process. Like my biggest fear as a scientist was never that I wouldn't find the answer, but more that I wouldn't be able to ask the next interesting question in order to have a lab that's sort of, sustainable and moves forward and has interesting things to study. I was, the fear is sitting in a lab and being like, well, gosh, we finished that. Now what are we going to do? So that love of science.
Yeah, that's so important. And when you had mentioned, having it be okay to fail in the lab, I was also going to ask Claris, because when I was in high school, you have the lab manual and you have every single step set up for you, and then you get the exact result. And I'm just wondering, Claris, what's your experience? And then how, like, there must be a really steep learning curve when it comes to doing it on your own.
Yeah. Like you said, we have every step laid out, and usually, if something doesn't work, it's because you did it wrong. It's not that, we don't already have the answer, or it's like we broke something on accident, But so yeah, I personally think failure is a very important part of science that I haven't really been able to experience myself yet. And I look forward to doing that when I graduate and do have more lab experiences.
Well, I mean, it's definitely always the first question, like, oh, the experiment didn't work. Did you make all the reagents properly? Right? Like that's part of the process. And sometimes you're just not going to do it right. I mean, it just happens. I had a friend in grad school and she would track in her experiments, she would track a radioactive molecule. So she would add this molecule that was radioactive and she would track it through a variety of chemical processes. And it took 15 hours. And, one time she did the whole experiment. She was like, I forgot to add the radioactive molecule. Or one time I made a peptide and I purified it in these test tubes. And then I went to the sink to dump out the ones it wasn't in. And I dumped out the ones that have my peptide. I'm like, and they right at the sink. I'm like, oh, they're wet.
Yeah.
So that's always part of it. But yeah, it certainly goes to show, like when you're in a chemistry lab and you have that lab manual, that's like, I mean, that took years to develop, decades to develop a lab manual that could be that specific. And then it's like, well, I don't know. I don't think that's very much fun.
No.
I mean, that's like cooking, but you don't get to eat the product.
Yeah. No, exactly.
It's kind of, it's pretty, change color.
Yeah. When students come into your lab, are they expecting more of that type of thing? Are they expecting more guidance? Is it like a harsh reality when they realize that they sort of have to start it on their own?
Probably in the beginning, before I realized how communicative I needed to be, right, with my students, about like thinking about how I operate and how all the things I'm dealing with, because I'm trying to teach all my classes and I'm trying to run a lab and I'm trying to write these grants, you're trying to shuffle. There was probably that piece. For them to get used to that, and for me to get used to the fact that I needed to make sure that was clear. But I think it got better over time.
What's a day in the life for you? Because you have all these things you're working on. Like, what is your general day?
I mean, it's definitely different now as an associate dean. I would say as a chemistry professor, if I was just a chemistry professor and I wasn't like department chair or anything like that, I'd probably come into work, check a bunch of email, try and respond. Then I'd go teach class for like an hour and a half. Then I might have to grade, or I might be prepping my next classes. I did a lot of hands-on work in the classroom, so that takes a lot of time to prep. Or to revise curriculum that I want to be like, okay, I didn't like the way I did this activity last year. I'm going to revise it for next year. Then I'd have students in and out of the lab. Sometimes if I'm teaching lab, that could be like, that's like a 4-hour experience. I might also be teaching an advanced lab down the hall while I've also got research students in my lab trying to do experiments. It's very much like very, very many competing needs all the time. So yeah, it's a little bit like, you have to focus on things in order to do really well. And research in particular, like you've got to be able to sit down, turn everything else off and get in the zone, especially if you're writing a grant or something like that. So summertime was really important for that type of work, because then I could really focus without students coming to office hours or swinging by, I need help, or can you sign this form, that type of thing. But it's fun. Every day is different.
Yeah. It's always a surprise. Maybe.
It's always a surprise. Luckily, nothing super bad has ever happened, knock on wood.
What would you say? What is your advice to people that are interested in chemistry, or also who are struggling in chemistry? What would you say to them?
I would say definitely, if you're interested in chemistry, you should pursue it. You should not let whether or not you think you're good at it be the determining factor. Too many students I've seen think they're not good at chemistry for whatever reason, and it's just not true. So you have to seek out that sort of mentorship to get better. And I think for students that are struggling, the most important thing is to go see your professor as often as you possibly can. I know that's really hard on students. It can be very scary. I never thought I considered myself intimidating at all, but apparently like it's a big deal. I know students think that if they ask a question that their friends in the class will think that, you know, they don't know anything, or that the professor is thinking negative things. And, you know, maybe they are, but in the end, it doesn't matter, right? All that matters is how you're doing. And if you're in something that you think is just super interesting, if it's hard, you just keep asking the questions and getting the help and seeking out the people that are going to support you in that. And normally in college, there are lots of avenues for that. It might be your classroom professor. It might be another professor in the department. It might be an upper division student or a graduate student, right? Like you can email me, right? So, you know, you can find the people that are going to provide you that strong support network. But it's so hard sometimes when I see students that are quitting because they think they're not good at it. It's like, and especially some of the students that are, you know, A students in the classroom, like 3 midterms and a final, and they're killing it, walk into lab and it's a whole nother ball game, right? I mean, that says something about how we're teaching chemistry, but a lot of students really excel in the lab. And then that's how they actually start learning the chemistry from their classes, right? Is because they're in the research lab and they're like, oh, this is what it is, right? Because they're physically doing it. It's not all written out in the lab manual. You know, they're figuring it out from scratch, and then it makes a lot more sense.
And for people that are scared of lab, I was one of them.
Oh, yeah.
What do you say to those people? Because sometimes that anxiety can just be crippling, even if you're doing really well in the classroom.
Oh, yeah. I was scared.
Mm-hmm.
But, you know, I think it's kind of the same thing. You know, one of my colleagues who, not a professor, but they worked in a department, and her daughter was getting ready to take chemistry lab, and she was terrified of lighting a Bunsen burner, which I too used to be terrified of lighting a Bunsen burner. Right. And so I'm like, well, have her come in with me and we'll practice. Right. And so, you know, she would come in, I'd be like, okay, we're just going to practice. And sometimes, you know, again, it's hard, right? Because you know, not all professors are the same. And if you told one professor, I can't light this Bunsen burner, who knows what terrible things they might say, right? Mm-hmm. But there's got to be somebody, right, who can help with those things. And I think being upfront and honest mm-hmm. is one of the ways to do that. And that might even mean going to like the tutoring center, right? And maybe they know somebody who tutored chemistry who could help you. But yeah, it could be debilitating for sure. And plus too, if you have a lab partner that wants to do everything and then you don't get the experience, and so that doesn't help either, right? It just sort of builds on itself.
I completely agree with that. And it seems like you've been able to, like, as you're integrating the different majors and the different types of education, it seems like you've been able to kind of foster an environment that is really, really encouraging. And what do you think it is in your curriculum that is more inspiring and encouraging than in like traditional curriculum?
Well, I think the pieces about the curriculum are that students, especially in my later years of teaching, once I sort of changed what I was doing, is that we spend longer on specific topics, right? More hands-on, more time for students to really grapple with questions that they could access in different ways, right? And more time in class, spend a lot more time in class talking with students, as opposed, I don't lecture anymore. So I try to let the students talk. I try to listen to what the students are saying, because then I can tell where the problems are. So I think that helps a lot. I think too, like part of it is that people don't expect me to be good at chemistry. There it is. Right. So I think that helps other, at least female students, but that are like, oh, well, if she can do it, and she smiles a lot and she has a high pitched voice, or at least when I don't sound like I've been coughing forever, maybe it's not quite so terrifying, but it goes back to the whole diversity and representation issue in STEM, right? That like, if 20% of the chemistry department are women and or underrepresented minorities, that doesn't do much for students that are looking for somebody to help them who looks like them or who can understand more about where they've been.
Yeah.
So,
I think we need more people with purple hair in chemistry. I really do. I for me, I have in my department, it's a lot of male staff, and my way of like standing up against that is that all of my PowerPoints, I put a pink background on them, every single one. And the information is still really great, and I still do fine. And it's just my way of like, it doesn't matter what color it is. It doesn't matter if we're girls. Like it's just so important to me that we're starting to see more people that are taken seriously, no matter their gender, what they look like, anything like that.
Well, it's interesting too, because like if students ask me a question in class and I don't know the answer, I'll be like, I don't know the answer. I'm like, oh, that's a really good question. I'll have to go think about that longer. Let me work on it for a while. But I have colleagues, I know people that lie. And I'm like, and it's a completely different perception, right? I can imagine too that a large proportion of the class, if I say I don't know the answer, thinks, oh, well, she doesn't know the answer. She's not a very good chemist. And it just means, no, I'm actually just being honest. And none of your faculty know all the answers and they're just not telling you. So I don't have time for games like that.
No. Yeah.
Not at all.
What was your experience with, I guess, representation or misrepresentation in like all your education in chemistry? Like growing up, did you have some sort of figure to look up to? Maybe not with purple hair, but
my mom was a school principal. So, she certainly, did her own, paved her own path. But she went to college, so she's educated, that type of thing. Where I went to school, back when I was at Harvey Mudd, there were like 130 students in my class and 28 of them were women. So it was like a 5-to-1 ratio. So, but I was I don't know if naive is the word, to recognize the lack of diversity. I think because at the time, I was always just sort of going with the flow. I didn't think about things, like I wasn't a big planner. I wasn't a big worrier about where I was going to end up next. That's my own privilege, right? I just knew that it'd be fine. But, later in my career, I think I started seeing it, especially at Sonoma State and in the academic conferences at this level, just in general. I've had colleagues make comments and I'd be like, that seems inappropriate. Like, why did you just say that? And then I start seeing it. And then especially, I think maybe the biggest thing was I was on a search committee once. And I walked into the room and there were like 9 people on this committee and they were all white men. And then there was me. And I was like, and then I sat there like while we're talking about what we were going to do. And I was like, oh my gosh, this is how a lot of my students feel in class. I'm all focused on the fact that I'm the only woman in the room. There are no persons of color. And I'm sitting here trying to at least rep for women. And I'm like, my students are sitting in class thinking about these things instead of being able to pay attention and to participate and engage fully in the chemistry. And then I think that moment I was like, I gotta really start figuring some stuff out and really reflecting on what's going on. So, I had a really good freshman chemistry teacher who was very inclusive and very supportive. Like actually all of them were, to be honest, like just very supportive, no matter how well or how poorly you were doing in class. I knew I could always count on them. And so I was already trying to model that behavior But I think later now, like I've changed a lot to try and be more inclusive so that maybe I can be that person for somebody else.
Yeah. I think people underestimate the importance of having a really incredible teacher, because I know there are so many subjects that I've personally been turned off of. And maybe Claris, I'm not sure if you feel the same way, but there are certain things where I've been like, yeah, absolutely not ever looking into that, because I had a teacher that I just really didn't like, or that I thought was mean. And then there are certain people that I just loved and that have made my experience in subjects I thought I would have hated really, really good. I think it's so understated, the importance of really good educators.
Yeah. I think too, like when every department has the mix, right, of all of it. So sometimes students would come to my office and they'll be upset about, oh, this teacher did this and that or whatever, and it'll be like, okay, well, you're going to meet a lot of people in your life and they're going to be challenging to you to interact with for a variety of different reasons, right? So how are you going to get what you need and be successful in this class despite the additional hurdles, which are unfortunate that you're going to have to face, right?
Yeah.
Because sometimes there's just nothing you can do about that, right? That person teaches that class and they're the only one.
Have to persevere.
How are you going to survive? What survival skills are you going to develop, to try and flip it in terms of the approach? Mm-hmm. Which, I mean, certainly, right? That's the hardest part of any job. I can tell you that right now, is playing well with others. It's not how much chemistry do you know, how well can you make an academic plan? It's how well can you communicate and interact and work together. It's fascinating.
For you as a professor, like, what is the best and worst thing, I guess, and what motivates and inspires you every day?
I mean, I think the best part and the things that have motivated me the most are is partly when students that have been struggling, like, figure it out, like they start making progress. I've seen students turn it around from 20% to getting a B or a B plus in the end. Like, I think that's awesome. I mean, personally, for the students that have worked in my lab, I know them the best, and watching them go on to do amazing things, that's incredibly satisfying and fun, because it's kind of like I had one student who went to graduate school at UC San Diego and worked for the same person I worked for, which was pretty cool. So just watching them go out and do science and be happy, I mean, that's the bomb right there. And that's what I do now, only I do it for multiple, not just chemistry. So I help faculty in other departments with their curriculum and how they support students. And then I help the students that are really upset. So yeah, it feels good to help students navigate college, especially at Sonoma State, right? We have a lot of first generation students. We're a Hispanic serving institution. So we have a lot of opportunities to really make a difference here at Cal State. So I really, really like that part. I went to a private college, so it's my own privilege, right? It's a little bit different experience than what we have at Cal State, but the education, totally the same.
Yeah. Yeah, Sonoma State is a really great school.
Yeah, I love it here.
Yeah.
Plus, they let me have the purple hair, right? At least nobody said anything.
Yeah, I think it's great, and it's a great area to be able to go to school in as well.
Yeah.
And I have one last question for you as well, which is, you mentioned that the sign of a good scientist in a good lab is always having questions and things you're interested in. So if you were given all the funding that you wanted, what are the top few things that you're super interested in?
I mean, we have so many open questions. I have so many experiments still to do. Yeah. I have this project with metals and looking how you can add metals to these bacteriocins and they sort of work together to become an even better antibiotic. I really want to answer the question about, if we put the two molecules together, can we recover the natural molecule? And what happened? If both molecules are active on their own, did we change who they can target? Right? Because remember, when you have the whole molecule, it's very specific which bacteria it will kill. But now, like, will the front kill anything? Do we make some sort of broad spectrum antibiotic, right? Like, there's so many questions. And then, there's 45 amino acids. We could change each one. I mean, it just goes on forever. Yeah. So yeah, there's a ton. But every experiment takes a really long time.
Well, I think you've officially gotten us interested in Listeria.
Oh, good.
So thank you so much. If you are listening to our podcast and would like to know more about all things chemistry, please go ahead and visit our website at chemistrytalk.org. Thank you so much and see you next time.